Good morning, everybody. Move this out of the way. Come on, everything's a bit slow today. Dum dum. All right, so where were we at? All right, so we finished up with that. Let's go back up here. This bit. So does anybody have any questions about the lac operon? Most likely in particular, the role of cyclic AMP in modulating the lac operon. Now's a good time to ask by chat or, in person. Does this make perfect sense? Or is this 100% confusing? Or is it somewhere in between? Kind of like bacterial gene expression. Inversely. Oh, well, I can't even spell proportional. So do you mean how does, like, what's the mechanism by that relationship between glucose and, oh, that's a really good question. Buried deep in my mind, I think I know the answer, but right now I don't, is the honest answer. And no, you don't need to know that. I do need to look it up, so I know it, in case someone asks me that question again. Yeah, I knew that once, but it's buried too deep to be able to kind of pick it out on a whim, unfortunately. Caitlin, she asked how the mechanism behind the relationship between cyclic AMP and glucose. And yeah, it's like, it's in there somewhere. I just can't get it out. But anyway, you don't need to know that. All you need to know is that cyclic AMP is what is sensed. Oh yeah, so the catabolite activator protein is kind of analogous to LAC-I. Get back, here we go. The repressor protein, in that it's always produced at a low level, so it's always there, right? It's encoded by another gene, as is LAC-I, but that gene doesn't have any kind of transcriptional control of it. It's always produced, but at a low level. So again, remember the context of kind of gene expression, right? You need the CAP protein or catabolite activated protein to be present all the time, just as you need LAC-I protein to be present all the time. And then there's other things which change how that protein functions, right? So the activity of that protein is controlled by other things, right? It's not controlled at the level of transcription. Whereas the lac operon, that is controlled at the level of transcription. So then you need to have a means of turning it on and off and, controlling the level of it. So there will always be a subset of genes which are always expressed at differing levels, right? Depending on the strength of their promoters. And there'll be a whole nother subset of operons which are under transcriptional control or regulation, right? Because they're either not needed all the time, or the amount to which they're needed varies. Does that make sense? Kind of like two classes of genes, essentially. Yeah, so LAC-I and the CAP are definitely part of that kind of always on. They're called constitutively expressed because they're always expressed genes, whereas the lac operon and trip operon are conditionally expressed, I guess is probably the best way of putting it. So depending on conditions and context. Yeah, good questions. Other questions? I will read up on Psycho-CAMP, although I have a sneaky feeling from memory that it's a bit of a rabbit hole, to be honest. I've dabbled a little bit in metabolism, and it's a bit of a swamp. It's like again, you can't get out again. All right, so we're all good with the lac operon because we're going to move on to the trip operon forthwith. And the trip operon is kind of crazy. Pardon me. All right, so let's move on to the trip operon, right? And so the trip operon, let's get this up in chat. The trip operon encodes everything needed to. You can ask Stephanie. While I type this out, see if it comes to mind. Then it's better to cover it here and now. Oh, well, can't help you with that. Right, yeah. So the trip operon encodes everything needed to make tryptophan, which is an amino acid, from chorusmate, which I have absolutely no idea what that is. It's just a compound of some sort. Okay, so now we have a very different context. So with the trip operon, the lac operon, sorry, we don't need lac operon expression if no lactose is present, right? So it's one of those things which is generally always off unless the bacterium needs to turn it on. Now, tryptophan is always needed to varying amounts, right, in translation, protein production. And so you always need some, a level of tryptophan, otherwise translation ceases, right? And actually, kind of like a little bit of a diversion aside, an awful lot of antibiotics actually target translation in bacteria, right? Because if you do that, the bacteria can't grow essentially, and eventually it will die, right? So translation obviously is a super important part of bacterial life or cellular life. And so the bacterium always needs tryptophan, but it also has to balance that need with the cost of making all of the components to make tryptophan, right? So it's kind of like cellular homeostasis again, right? That concept that I talked about a little bit last class, I think. We want enough to be able to do what we need to do, but we don't want to do any more than that because that's wasted, right? It's using energy that could be used to do other stuff, reproduce, fight off other bacteria, stuff like that. And so this context is very different from the lac operon, right? Now we have the context where this operon needs to be on all the time, but we need to dial it back when it's not needed, right? And so that's a very kind of different context. And as with the lac operon, right, glucose essentially feeds back via Psycho-CAMP to dial back the lac operon, right? So it's kind of a negative feedback, right? So the product, glucose, turns down the expression of the operon which generates it. The same applies with the trip operon. Tryptophan, and I'll go through this in more detail in a little bit, just want to cover the concept. Tryptophan is what is used to kind of feedback and turn off the trip operon, right? So here's our feedback loop again, right? And this is part of how this balance is achieved, essentially, between needing tryptophan and not wanting to spend a lot of energy on making all of those proteins to make tryptophan. Okay, so we have the same kind of deal with the trip operon, right? But it's now always on unless it's turned off, right? So now we have a repressible system. That means it's always on unless it's turned off or repressed, right? So again, remember the outcome of the action of these things. In this case, the repressor turns off the trip operon, so it's a repressible system. And it looks in some ways very similar to the lac operon, right? So we have a bunch of structural genes. These encode the enzymes that turn chorosmate into tryptophan. I think my puppy's been a pest. Right, so you can see here, these are the enzymes which turn each of these individual intermediates into the final product, right? So obviously you need all of them to be expressed at the same time, otherwise you don't get tryptophan. We have a promoter where the RNA polymerase binds, and we have an operator where the repressor TRPR binds. So in trip operon, TRPR is the repressor. Aha. But unlike LAC-I, it's an aporepressor. Anybody remember what an aporepressor needs in order to be able to function, to be able to bind DNA? A co-repressor. What do you think the co-repressor is in the case of the trip operon? Given that we have a negative feedback loop, it's controlled by the thing it produces. Yeah, it's tryptophan, right? So only in the presence of tryptophan, oh dear, here come the dogs. Little one is in gonzo mood for some reason. She's a little furry pest, particularly to this one. So we have the TRPO operator, that's where TRPR binds in the presence of tryptophan. You just calm down. Sorry, a little distracting. And so far, so kind of similar to the lac operon, right? But now we have an extra region. This is called the leader region, right? So TRPO and the operator and TRPR, obviously plus tryptophan, are all involved in the big on-off nature, right? TRPL, leader region, equals the fine tuning. Hey, Stop it. Which is, in the case of the trip operon, is called attenuation. Right, so this is the bit that's analogous, even though the mechanism is completely different, analogous to the cyclic AMP catabolite activated protein, that kind of intermediate level of expression, that fine tuning. Okay. So yeah, that's pretty much all there. Oh yeah, also in that leader region is an attenuator sequence. And obviously, as you guess, this is involved in the attenuation of transcription. And to attenuate basically means to cut short, is the, I think that's actually from Latin, not entirely sure how, but attenuation means to cut short. So bit of language there. Okay, so those are the two main parts, right? So the first part is pretty straightforward. I'm actually going to skip the words and go straight to the image because you can read those later if you wish. When you don't have enough tryptophan, and obviously this is not good, right? Because if you don't have tryptophan and you need to make a protein with tryptophan in it, you cannot, right? Generally speaking, I think bacteria make all of their amino acids, right? They don't get them from their environment the way we do. I think we can only make, I don't know, around 10 of the amino acids that we use for translation, making our own proteins. And so we actually have to get a lot of ours from the food that we eat, like beef. So anyway, if tryptophan is low, that's a bad thing, right? And so we really need, as a bacterium, to make more of it, right? Otherwise, translation comes grinding to a halt. So in the absence of tryptophan, tripoperon is expressed. And the reason why it's expressed is because there's no tryptophan to bind to TRPR, and so it has the wrong shape, right? It is unable to bind DNA, and it doesn't, right? So there's nothing bound to the operator to prevent RNA polymerase from transcribing the tripoperon, right? And as you know, we've learned in lac operon, if there's nothing there, that operon will get transcribed. Right? And in this case, I think the TRPR gene is somewhere else in the genome, right? It's not next to the tripoperon. Okay, so that's relatively straightforward, right? No tryptophan, essentially the co-repressor, the apo-repressor TRPR is unable to bind, and so we get transcription. Now, when you get transcription, you get translation, chorismate, provided there's enough of it present. And actually, that must be something they get from their diet, I think. I have to look into this more. Chorismate gets turned into tryptophan via a series of steps, right? All catalyzed by the products of the tripoperon. Okay, so now, presumably tryptophan is being consumed, right? Because it's needed in translation. But over time, you'd expect tryptophan levels to start rising, right? Now, when they get high enough, then there's enough tryptophan to bind to the apo-repressor TRPR and turns it into the correct conformation where it can actually recognize the operator and bind to it. And that shuts off expression pretty tightly, actually. Right, this is not a leaky system. And so when TRPR is bound, even if RNA polymerase is able to bind the promoter, it cannot get through it, and so there's no transcription. So this is the negative feedback loop, right? The operon that encodes the things to make tryptophan is negatively regulated or turned off by tryptophan. Okay, how are we doing? All right, anybody else chance to pipe up? Give me time to drink some of my coffee. It is Friday. Woo! Fun times. Weekend of freedom, blazing around, lying in until at least 7 o'clock in the morning. Yeah, Caitlin. For now it is. Do not be fooled. Okay, so the on-off switch for the trip operon is pretty straightforward, you're right. The attenuation is not. So I want to go through this, in a pretty measured way. Okay, so let's use this time to have a little bit of a kind of think back to transcription and translation. What is one of the characteristics of prokaryotes? Pardon me. With regard to transcription and translation, how does it differ from eukaryotes? They're coupled. What does that mean, Stephanie? What do you mean by that they're coupled? They happen at the same time, right? And so that's right, Melody. Well, essentially, as soon as there's enough RNA transcribed, you can begin translation. And so actually buggeration. I don't have that up. Hang on a second. Where is that? I think it's in this one. Get out of it. Where are we at? There we go. Exactly. So you're both 100% right. Essentially, you can start producing protein. You don't actually show the protein here, but it's being made. You can produce protein even while the mRNA is still being made, right? So that's one kind of real central part of attenuation, this coupling of the two. And I'm pretty sure I mentioned that when we talked about this, that we'll be covering it when we get to the trip operon. Okay, so keep that in your mind. Coupling of transcription and translation, right? They happen at the same time because they happen in the same place, right? Okay. Another question. What is required or what needs to be formed for transcription to terminate in prokaryotes? This is probably a little bit trickier of a question. What's one of the key features or things that RNA can form that has to have, yes, a loop, a hairpin loop, to be super specific. And that's in the mRNA. And that is equals transcription termination. Right, so if we kind of go back here, and actually it also occurs to a lesser extent in row-dependent transcription termination. That's more about slowing it. But here, this these inverted repeats form a loop. Okay, so these two things if I can spell transcription. So the coupling of transcription and translation and the formation of secondary structures, hairpin loops specifically, in mRNA equals the mechanism of attenuation in the tripoperon. Yeah, spell all that right. Give myself a pat on the back in a little bit. Okay. All right. Everybody with me so far? Because it gets way crazy after this. All right. Hold on to your seats, so to speak. Okay. So in the leader sequence, right, this leader region, there exists a series of inverted repeats. Right, in this region, go on, just here, right? And there's also an attenuator sequence, right? The attenuator sequence is essentially a hairpin loop that can form and disrupt transcription. So that's one aspect of it. So the leader region, oops, is transcribed first, and this is the really, really important part, starts to be translated before the structural genes are transcribed. Okay, so we've got transcription and translation, which are coupled, occurring before the RNA polymerase can move into the structural genes. What is tryptophan used for? What do you need tryptophan to make? It's not a trick question. Making proteins. And we're going to be making a protein, a short, nonsense, like little bit of protein that isn't used for anything, but is involved in the attenuation of the tripoperon. Okay, can you see where I'm going with this? Right, so the product of the tripoperon, tryptophan, is needed in translation. And we're going to be transcribing and translating a small protein before we get to the structural proteins. Now, if you're a betting bunch of people, who would bet there's a couple of codons for tryptophan in that leader sequence? I'm not much of a betting man, and I'd bet on it. What do you want? Sorry, obligatory. Can someone unmute their mic and talk to her? Hey, Pepper. Pepper.
Pepper. Oh, sorry, my dog's barking now.
She was too preoccupied with the trash truck that was outside, unfortunately. Anyway, she's helping keep my foot warm. No, it's all right. They don't bark very much, apart from this batty creature. And she just goes off when people walk around upstairs. She thinks like the house has been invaded by for some reason. I don't know why. Okay, so again, kind of like trying to build up to this. We have a short bit of RNA that's transcribed, the leader sequence. that leader sequence is translated, right, before the structural genes are. There's a possible attenuator sequence in there. There's a possible hairpin loop, and there are 2 trip operons. There you go. Fingers aren't as flexible as they used to be, right? All of those things are involved in attenuation. Okay, so I'll write those down. 1, leader sequence is transcribed and translated before the structural genes are transcribed, right? 2, I type in tryptophan. Tryptophan is required for translation of the leader sequence. Oh, hang on. Yes, that's exactly right, Stephanie. The mechanism of how it works, though, is a little tricky. So that's why I'm trying to build up to it. So all the kind of the context makes sense. But yeah, you've got it in a nutshell. How that works, though, is actually not trivial, essentially. Now, where was that? Tryptophan is required for translation lead sequence. Yep. And in the presence of tryptophan, the attenuator sequence, which is basically at the end of the leader sequence, forms a terminator loop and stops transcription. So in a sense, what we're doing here, or what the trip operon involves, is it uses tryptophan as, well, it uses translation of the leader sequence as the detector for tryptophan levels. Right, I'd say this wasn't easy. And so essentially what we have is 2 possible conformations of the leader sequence. Okay, so let me just blank off this. Oh, come on. Okay, so here is, or over to the left, is the trip P and trip O. All right, so there's not enough tryptophan to turn off the trip operon. Okay, that's to be quite a lot of it for the trip operon to just be completely shut down. And we have the structural genes over here, right? So this is trip E, and then there's D, C, B, and A, kind of going to the right. So this essentially is in between, right? We also have a ribosome binding site, Shine-Dalgarno sequence, and a start codon, sorry. Okay, so this sequence can be translated. It's called the UTR, but that's a little bit of a misnomer because it will be translated in part at least. We also have 4 different regions. These regions can base pair with each other. And so there are 2 possible kind of conformations, right? Oh, there's a Q, not W in sequence. has 4 regions which can get out of it, base pair with each other. So there are 2 exclusive or mutually exclusive conformations. There might be an I in there instead. Okay, so we can have version 1 equals 2 and 3 bound together. Okay, now if 2 and 3 stick together, 3 and 4 can't. Okay, this is really, really important. Version 2 are essentially either, always have to be kind of careful here, basically 3 and 4. I've got to be careful because it's important when we talk about the mechanism later. I don't want to confuse you. 3 and 4 are bound together, and that equals the terminator loop. And see the U's at the end? That will slow down RNA polymerase. And if the terminator loop forms, it will kick RNA polymerase off of the DNA. Okay, so we have these 2 different versions. Now the key is how one version is chosen over the other. If one forms, the other one cannot, and vice versa. All right. And the key to this are these 2. Where did that circle come from? Ah, badges. I lost it. Oh, here it is. It's being lazy. Oh, well, that didn't work very well. All right, sorry. What did I just do there? Something I didn't mean to. I'm just trying to get around having to always empty the fill. There you go. Anyway. So the key way in which those 2 versions are kind of, one is chosen over the other, is down to the presence of these trip operons. Okay. So these trip operons require tryptophan to be present to be translated. Okay, and so what we're actually going to see happen is the ribosome is the thing that chooses, essentially, not deliberately, but by its presence, version 1 versus version 2. I'm actually going to skip on to the next images because they're a little bit easier to kind of understand. Is that okay with everybody, or do you want to see what these different formations can look like? I don't want to jump ahead too much, but I also, well, I trust my judgment. Woohoo! I've never had a student say that. Thank you, Stephanie. I'm not sure if that judgment, that trust is wisely placed, but okay. So remember, transcription and translation are coupled. So we have, and I'm going to blank this off to make life a little easier. I also want to make it a little bit bigger in a second. Okay, that's smaller. There we go. That's better. Bit of a fuzzy image, but it'll do. Okay, so RNA polymerase comes in, begins translating the leader region. Remember, this is this series of 4 regions which can form different conformations. RNA polymerase quite happily chunters through, right? Now, as soon as there is enough mRNA present, a ribosome can bind to it and begin translating it, like so. Okay, so the ribosome goes dunk, 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 2, and the whole thing is moving along, right? This is really, really important. These 2 things are progressing at about the same pace. So as the ribosome starts translating region 1, RNA polymerase is transcribing region 2. Okay, and remember, these trip codons are in region 1. Okay, so here's a question for the class. What do you think will happen in terms of translation when the ribosome gets to these trip codons and there's no trip amino acid or tRNAs with tryptophan available? Tryptophan levels are low, there will be no trip tRNA. So what's going to happen in terms of translation? What do you think the ribosome will do once it gets those trip codons if it has no trip tRNA coming in to add tryptophan in that spot? Exactly, it will stop. Now, will the RNA polymerase stop going, even if the ribosome is stationary? Because they're not linked. No, it wouldn't, right? So now the ribosome is going to pause on region 1. And remember, the ribosome is what dictates which regions pair together. This is completely and totally the whole center of this process. It's all about whether or not the ribosome stops. Okay, so the ribosome is going to pause, but the RNA polymerase is going to continue on. And it's going to keep on going, right? But now region 1 is not available to pair with region 2 because it has a honking great big ribosome parked on top of it, right? But region 4 is also not available because it hasn't been made yet. And so what you then get is region 2 and 3 form. Right, so back up in the chat, said that these 2 versions are mutually exclusive. So if region 2 and 3 form a loop, doesn't affect the RNA polymerase because RNA polymerase is already gone, right? So if region 2 and 3 form, RNA polymerase just keeps on going. And it transcribes into the structural genes, and then you get trip operon expression. Okay, so just to kind of go over that again, because this is not a trivial thing to get at all. Let's kind of scooch. Okay, so transcription begins. Once enough mRNA is produced, translation begins. And by that point, transcription is already kind of proceeding further through the leader sequence. When the ribosome gets to the trip codons and there's not enough tryptophan because trip levels are low, it stalls, right? It cannot continue because there's no amino acids to add to the polypeptide, right? This little protein here, this leader protein, what it is not, doesn't matter. It doesn't do anything. It's merely the means by which tryptophan levels are detected, right? Those intermediate tryptophan levels, right? The fine tuning off and on again. RNA polymerase, however, is not affected by the ribosome stalling. And so it continues onwards quite happily and produces region 2 and 3 before region 4. And so then 2 and 3 are free to bind and form this loop, and 4 is not able to, right? Because 4 would normally bind to 3, but 3 is already bound something else. It's like, I'm not going to talk about, boyfriends and girlfriends. That's totally inappropriate. Plus, I have 2 girls, and so I cannot talk about boyfriends at all, at least for another 10 years. So if that forms, transcription just continues straight on through, right? And the trip operon is expressed. Now, what happens when, as a result of the trip operon working, tryptophan gets produced, right? A bit of it, not a lot, right? So trip R still isn't binding, so we're still going to have transcription. But now, let's start the same process again. Right, and now you'll see how the ribosome and translation of the leader sequence is the sensor that detects tryptophan levels. So the same thing happens again, right? RNA polymerase binds because there's no trip R bound, or trip R and tryptophan bound to the operator. Region 1 is transcribed. So far, so similar. Region 2 begins to be transcribed as region 1 begins to be translated. But now, okay, now we have sufficient tryptophan so that that ribosome does not stall. Okay, so we have enough tryptophan that there's tRNA comes in and the ribosome continues on. Now, what do you think will happen when the ribosome is over region 2? Is region 2 and 3 going to be able to bind together? That's a big no, right? And so if region 2 and 3 can't bind together, that means region 3 and 4 can. And again, RNA polymerase is far enough ahead that ribosome is going to be on region 2 by the time the RNA polymerase is paused, right, because of those U's, or it slows down. And the terminator loop forms, and that kicks the RNA polymerase off and stops transcription. So the whole mechanism of attenuation really comes down to those 2 trip operons. That and the ribosome are the sensor that detects tryptophan levels. And as you can imagine, this is a pretty sensitive one. So essentially, these two slides are different conditions. So this is what happens when tryptophan levels are low, right? So you imagine, like, once the trip operon starts really gathering steam, right? So it's being expressed, but there isn't a lot of tryptophan being produced. There's still not enough tryptophan to be on the tRNA to allow this leader region to be translated, right? The ribosome stalls here. And so obviously you need more tryptophan so transcription continues, because the formation of this 2-3 loop. Now, once trip levels start going up, you can see that, well, maybe there's enough for 1 trip tRNA. Okay, so the ribosome still stalls. A little bit more trip operon expression. Now we have enough for 2 trip tRNAs to be present for translation of the leader sequence. Now that ribosome doesn't stall, right, because it has enough trip tRNA to continue, and it continues into region 2, right? So this version cannot form when there's enough tryptophan present because there's a ribosome on it. And so the RNA polymerase keeps on going as it does, and it transcribes region 3 and 4 And now region 3 and 4 can form the terminator loop, which kicks RNA pol off. You know, no trip. Sorry, we've gone over a little bit, but it's worth kind of, you know, making sure this is all clear. So now there's no tripoperon expression. Tryptophan levels start dropping, right? And we start going back to the situation where there isn't enough tRNA to translate the leader region or leader sequence. So the 2-3 loop forms, no termination occurs, transcription of the tripoperon continues, and we get conversion of chrismate into tryptophan. Tryptophan starts going up, and we have enough to pause, not to allow the ribosome to continue, and pause the polymerase and kick it off with the formation of the terminator loop. So it's going to be constantly kind of flipping between these two levels. As long as tryptophan production is on, right, but it's still kind of being consumed, right? There's not enough of it to really totally turn the tripoperon off. So you can see that the tripoperon is most likely to spend the majority of its time in this kind of dithering intermediate state, right? Where there's enough tryptophan and then that terminates transcription. So we get tripoperon expression, and then we get more tryptophan and that terminates transcription, right? So it's constantly kind of floating around that kind of level of just enough tryptophan. All right, I've gone way over. So yeah, already covered that. Have a think about that, and we'll have a few minutes at the start of lecture on Monday to go over any questions that you have, same as we did today for the LAC-OPERON. Okay? But remember, all of those different points that I outlined in the chat are, I think at least, it's the way I make sense of it. So hopefully that'll help for you, but have a look through it and see how this all works. Bring any questions to class on Monday. All right. Have a lovely weekend too, Armando, and everybody else. And I'll see you on Monday.